Okay, class, today's lesson is about membrane transport. How do membranes achieve the goal of semi-permeability? How does it let some things in and some things out? Okay, so here's how it works. All right, so first of all, there's just simple diffusion. You guys remember diffusion is just going from high concentration to low concentration. Okay, so of course, as we mentioned before, nonpolar things like oxygen, carbon dioxide, okay, they go from high to low. So as a cell, this is intercellular, this is extracellular. As the cell uses up oxygen, there's always going to be more oxygen here than inside, and it can diffuse easily across this this nonpolar layer here, those fatty acids there. Okay, same thing. Carbon dioxide is produced inside the cell, so it can diffuse out easily. Okay, so that's simple. Diffusion, they call it passive because it doesn't require any energy and it goes with the gradient, always from high to low. Okay, same thing for water. Water actually uses these protein channels called aquaporins, okay, but they are polar on the inside all the way through. Water's small, so it can kind of fit right through there. So the process of water going through a membrane is called osmosis. Okay, so osmosis is specific for water, high concentration, low concentration. It's all diffusion. Okay. Now sometimes you need a little help, so facilitated diffusion is done for ions, proteins, glucose. Okay, molecules which are a little bit bigger, but they also go with the gradient. So they go with the gradient, so there's no energy needed. Okay, always go from high concentration to low concentration. So in a cell where you use up glucose, for example, okay, you're going to have more outside than inside usually. Okay, so glucose, I didn't draw this very well, but they're bigger, okay, but they use what are called these transmembrane proteins or carrier proteins. Okay, so they fit in here, and then there's a conformational change, and they actually transport them across. Okay, so they fit in, and they change shape, and it kind of, think of it as kind of squirting them across, right? So facilitated diffusion, you need these membrane proteins, but it goes with the gradient. Okay? Then we get into active transport. Active transport is two ways significantly different than diffusion. Okay, active transport, you have to have energy, okay, and you go against the gradient. Okay? So you're pumping things out or pumping things in to create some sort of concentration gradient. A really good example of that is what is called the sodium-potassium pump. It's why you're alive. It's what gets your nervous system, okay, energized or polarized. Okay, you have more sodium outside than inside your cell because there's a pump which pumps 3 sodiums out. At the same time, it pumps 2 potassiums back in. Okay, but you can see you can produce more positives outside than inside. So we say that the inside this cell has a negative potential. I think it's about negative 30 millivolts. Okay, but that's what keeps your nervous system ready to fire. Okay, you got that potential difference between inside and outside of the cell. And that's created by this sodium potassium pump. How it works, the sodium here, okay, fits in this, again, a transmembrane protein, okay, and they're transported out. Okay, once they get out, it takes 2 potassium backs in. So there's a conformational change in that membrane protein when the sodium locks into it, and when the potassium locks into it, it causes it to transport those molecules across the membrane. Okay, that's called a co-transport because there's 2 different materials being transported. If they're going in different directions, that's called antiport. If they're 2 different substances going in the same direction, that's called a symport. Right? So that's active transport. Again, it costs energy. Okay, so there's going to be some ATP used up here, and also it goes against a gradient. Okay, so it sets up this some sort of concentration gradient. Okay? When we study metabolism, you're going to see these. We're going to be pumping hydrogen outside. Okay, so stay tuned for that. Okay, then the last type, again, another type of active transport is endocytosis, okay, and exocytosis. Okay, so you can break down endocytosis into 3 parts. Phagocytosis, where the endocytosis means you're using that cell membrane to engulf it. Okay, so this is a membrane here, double membrane. It's say like a bacteria that's invaded your body. You you have white blood cells that go around and surround it and bring it in. Okay, so it comes in like this, and the membrane pinches it off, and then that bacteria comes inside the cell. Okay, and then of course lysosomes can fuse with it. Okay, and use their hydrolytic enzymes to break it up. Okay, and just to break it down, and so it's a good defense mechanism. Taking in solid particles is called phagocytosis. Taking in smaller liquid particles is called pinocytosis. Okay? And there's also what are called receptor-mediated endocytosis, where it's going to take in certain proteins in your body in a large-scale transport. Okay? Exocytosis is the same, but just going the other way. Okay? So, for example, after this does its work, a lysosome fuses with it, breaks down that bacteria. They want to get rid of it. So again, it will fuse with the inside part of the membrane, okay, and then it will expunge everything outside the cell to be dealt with extracellular. All right? So that's membrane transport in a nutshell.